So in the last video, I went through the structure of bases, nucleosides and nucleotides, and how you join together nucleotides to make RNA and DNA. We also talked about why RNA is so much less stable in the cell than DNA. In this video, I'm going to talk about the three-dimensional structure of DNA, as well as some more details about the central dogma. This is the structure of DNA, which I will talk about today, but once again, a relevant question is why should we care about this as future pharmacists? Well, for one, there is a class of anti-cancer drugs called anthracyclines. Doxorubicin is one example. These drugs have high affinity for the structure of DNA, and once they bind, they deform it. And what this does is it messes up the cell's ability to carry out transcription and replication. So in order to understand how anthracyclines work, we need to understand the three-dimensional structure of DNA and also the process of transcription and replication. As with a lot of things in science, it took a long time to figure out the structure of DNA. DNA was actually discovered in 1869 by Friedrich Miescher in the nuclei of white blood cells, but he called it nuclein, and he didn't know the chemical structure, just that it had a lot of phosphorus in it, and it wasn't protein. Following that period, up until about 1929, Albrecht Kozel and Phoebus Levene were able to isolate all the components of ribonucleotides and deoxyribonucleotides and determine how they fitted together to make nucleic acids. Levene also proposed that DNA was a polymer of nucleotides and that they were connected through phosphate groups. So you can see that they figured out almost all of the structural details about single DNA strands that I talked about in the last video. They just didn't know what DNA was for, what the order of the nucleotides meant, and what the three-dimensional structure was. Then, in 1944, it was discovered that DNA was the molecule in the cell that carried genetic information passed from one generation to another. This suddenly meant that DNA was a very important molecule, and figuring out how it worked took a new sense of urgency. A bit later in 1950, Erwin Chargaff discovered that different species have different ratios of adenine to guanine in their double-stranded DNA, but despite this, the amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. These findings ended up being known as Chargaff's rules. In 1952, Maurice Wilkins, Rosalind Franklin, and student Raymond Gosling were able to produce crystals of DNA strands and collect X-ray diffraction data. And from that they were able to tell that the phosphates were on the outside and that it was probably a helical structure. Then, in 1953, Watson and Crick published a three-dimensional structure of DNA that is consistent with the X-ray structure, what was known about bond lengths and angles, as well as Chargaff's rules. The most important thing that the structure told us that wasn't known before was that guanines specifically pair with cytosines through hydrogen bonds, and similarly, thymines specifically pair with adenines. In their structure, it is impossible to get other pairs, and so this suggested that if you have one strand of DNA, you can automatically determine the sequence of the other, and further, that the specific pairings may be how biological systems make copies of DNA strands when they are passed from one generation to the next. So the really important part of Watson and Crick's structure was that they figured out that in double-stranded DNA, specific pairs of bases are held together through hydrogen bonds. But what is a hydrogen bond? Well, let's start with the hydrogen part. We know, of course, that hydrogen is the simplest atom. The nucleus is one positively charged proton, and it has one electron shell containing one negatively charged electron. Now let's imagine that this hydrogen is part of a molecule. We think of covalent bonds as being two electrons shared by two atoms. In reality, the negative charge is not just in two places, as implied by this diagram, but spread out more or less equally along the length of the bond. We say that there is equal electron density along the bond. However, if instead of a carbon attached to the hydrogen we have a heteroatom, like nitrogen or oxygen, then the electrons are drawn towards the heteroatom. Why is this? Well, nitrogen and oxygen have a property called electronegativity. What it means is that electronegative atoms, such as nitrogen, like to draw shared electrons closer to themselves and away from the other atoms they are bonded to. The result of this is that the single proton that forms the nucleus of the hydrogen atom is more exposed, and therefore that area is more positively charged than it would be otherwise. Why is this important? Well, oxygen and nitrogen atoms have lone pairs of electrons, shown here as blue dots, which are of course negatively charged. And so there is naturally an electrostatic attraction between the positively charged proton and the negatively charged electron pair. What I'm showing with the dashed line is what is known as a hydrogen bond. It is not a covalent bond, but rather an electrostatic interaction. You can think of it as a bit like a weak ionic bond, because the partners are not charged quite as strongly as ions. A hydrogen bond has two parts. The part that donates the hydrogen, this is called the hydrogen bond donor, and the other part that contains a lone pair of electrons, which is called a hydrogen bond acceptor. So with that all in mind, let's see this in action in a base pair. You can see here that in the base pair of adenine and thymine, there are two hydrogen bonds, each one with a hydrogen bond donor and one acceptor. Here I've drawn red circles around the donors, and here are blue circles around the acceptors. And here is the other type of base pair with guanine and cytosine. Once again, I have circled the hydrogen bond donors in red and the hydrogen bond acceptors in blue. You can see here that the GC base pair has 3 hydrogen bonds, whereas the AT base pair has 2. That means that it takes more energy to pull apart, and in the laboratory, when we want to separate a double-stranded DNA molecule, it takes more heat to do so if the sequence contains a lot of G's and C's. These base pairs are called Watson-Crick base pairs, because Watson and Crick's DNA structure used these patterns of hydrogen bonds to explain the pairings. Another thing to notice here is that these pairings are specific. You can't pair the wrong bases together because then you would have hydrogen bond donors right next to each other and hydrogen bond acceptors pressed together. Even if you would squish these two bases close enough, there would not be any force or attraction between them and the structure would fall apart pretty easily. With all that we know now, let's look at the 3D structure of DNA in more detail. First, let's look at this GC base pair. It looks a lot like the two-dimensional chemical structure shown above, but the three-dimensional image I'm showing here was made using atomic coordinates from an X-ray diffraction structure, so we are pretty confident this is how the molecule would look like if you could take a photograph of it. And if we rotate the base pair, we can see how flat or planar the bases are. This is because bases have aromatic rings, and one of the features of aromaticity is that even though we've drawn some bonds as single and some as double, they are all the same length, making aromatic rings flat. Now if we look at an AT base pair, we can see that the overall shape is very similar. The bases are the same distance apart, and the glycosidic bonds come off the bases in both pairs at the same angle. This allows the overall DNA structure to be regular, even if it has a mixture of these different types of base pair. Let's now have a closer look at that regular structure in 3D space. You can see here that the bases are sort of stacked parallel to each other, but they are not right on top of each other. There is a bit of lateral displacement each time, so that the molecule looks a bit like a spiral staircase. A fancy name for this sort of twisted spiral staircase-like structure is a helix, and because this structure is made from 2 DNA strands, it is often referred to as a double helix. The reason for the displaced stacking is a phenomenon called pi-pi stacking. Aromatic rings contain delocalized pi bonds, and they like to associate or stack in these configurations. We can think of the bases in DNA as being like the parallel displaced conformation of benzene. So the structure of DNA that I've been showing you is what is known as B-form DNA, because there are actually a few other related three-dimensional structures that double-stranded DNA can exist in. But the important thing to remember here is that in cells, DNA exists as the B form, so we'll only consider that here. Now there is some flex in the molecule, but typically the distance between 2 base pairs is about 3.4 angstroms. And a complete turn of the spiral structure is 34 angstroms, or about 10 base pairs. As I said just now, the structure overall looks like a spiral, sort of like you would find in a spiral staircase. Now I don't know if you've thought much about spiral staircases, but there are actually 2 ways that you can build one. In this picture, when you go up the staircase, you are going counterclockwise, and when you go down, you are going clockwise. Now let's look at a different way of making a spiral staircase. In the picture on the left, you can see that when you go up, you are going clockwise, and going down would involve going counterclockwise. And just as there are 2 different ways you can make a spiral staircase, there are 2 different types of helical structure. But what do we call these types? Well, the name for the one on the left is left-handed, and the one on the right is called right-handed. Now, the problem with these names is that they are not descriptive or intuitive. You basically have to memorize what they mean. But how I remember this is that a standard screw is a right-handed helix. What this means is that the screw is tightened by turning it clockwise and loosens by turning it counterclockwise. And if you can't remember that, then there is a useful mnemonic, which is righty-tighty, lefty-loosey. This also isn't ideal because you have to remember that the arrow is above the screw as you are looking at it from the top, but it's the best that I can do. With all that said, let's look at the DNA structure again. It is a bit more difficult to see, but it is right-handed like the standard screw. If you imagine this molecule as a screw, with the top being the head, we can see that turning it clockwise is like tightening it, and if we turn anti-clockwise, we are loosening it. So the bottom line is that you should remember that standard B-form DNA is a right-handed helix. Now, one thing you may not have noticed is that B-form DNA is not exactly like a spiral staircase. In a spiral staircase, the surface area of the top of the stairs is basically the same as the surface area on the bottom of the stairs. We can think of the top and bottom of the stairs as the two surfaces of the helix. B-form DNA also has two surfaces, shown in orange and yellow here, but they are not the same size. The orange part is called the major groove because it has a larger surface area, and the yellow part is called the minor groove because it has a smaller surface area. But why is there this difference? Well, if we imagine taking a cross-section of the molecule, we can see that the glycosidic bonds joining the base's two deoxyribose units are angled towards the bottom. Consequently, the side of the base pair above the glycosidic bonds is larger than the side below the glycosidic bonds. These correspond to the major and minor groove respectively. But why is it important to understand about the major and minor grooves? Well, this base pair is a GC base pair, and we can also see what an AT base pair looks like. It turns out that proteins that need to identify specific DNA sequences interact with the major groove, because the pattern of the 4 hydrogen bond donors and acceptors depends on the identity and orientation of the base pair. By contrast, only one of the hydrogen bond donating groups is different in the two base pair types in the minor groove, and there is no way for the protein to tell which base is on which side. So the bottom line is that proteins that interact with DNA look for the regular B-form structure, and they generally interact with the major groove when they are sequence specific. Now, you may be wondering why we are going into so much detail into the structure of DNA, and one reason is that there are drugs that target the B-form DNA structure and alter it. They are called DNA intercalators, and they are used to treat cancer. One important class of DNA intercalators are the anthracyclines, which are all fairly similar in structure, just varying in these R groups. But the most important parts are these 3 rings, which form a flat aromatic structure. And the thing about flat aromatic structures like this is that they like to stack on top of each other, as we saw earlier with the nucleotide bases. And so these molecules like to slot between the bases in B-form DNA. But because there is not normally enough room for this between base pairs, the binding of anthracyclines between adjacent base pairs, called intercalation, distorts the local structure of DNA. Here on the right, we have a structure of B-form DNA with an anthracycline intercalating between 2 base pairs. And on the left, we have a normal structure of B-form DNA. You can see that the anthracycline has pushed the 2 neighboring base pairs further apart, and this has distorted the local structure. Because of this, proteins cannot interact properly with DNA, meaning that processes like transcription and replication are disrupted. So these molecules treat cancer by stopping cancer cells being able to make RNA to express their genes, and by stopping them replicating their DNA when they want to divide. Through these 2 actions, the growth of cancer cells are stopped. But because most of our normal cells also need to do these things, these drugs are also toxic to them, which causes a lot of side effects. Anthracyclines mostly affect normal cells that need to divide rapidly. So for example, we get a reduction in the number of white blood cells that are produced, and hair follicles stop growing hair. So we know that the sequence of nucleotides in DNA molecules holds information, but how much information needs to be stored in a cell? Well, the total amount of information a cell carries is called its genome, and the genome of the bacterium E. coli is 4,639,221 base pairs long. We know, of course, that the base pairs are on average 3.4 angstroms apart in B-form DNA, so that means the genome would need DNA with the length 15,773,351.4 angstroms. But what is an angstrom? Well, 1 angstrom is equal to 1 times 10 to the minus 10 meters, so we can multiply our length by this to get 1.577 millimeters. Now that might seem small, but the length of an E. coli cell is 0.002 millimeters, and so the genome is 788.5 times the length of the cell. And incredibly, the E. coli genome is just one molecule of DNA. But instead of a linear molecule, the E. coli genome is made of a circle of B-form DNA. The circle is called a chromosome, and it is drawn here to scale against this tiny speck here, which is the E. coli cell. So even though the chromosome is drawn as a ring here, if you zoom in, you would see that the molecule itself is still helical. But this is still a very big molecule to fit into a small cell, so how is this done? The answer is that extra twists are introduced into the molecule called supercoils, which help pack it into a smaller space. The DNA is already coiled into a helix, and so supercoil is basically a fancy word for coil of coils. Now, if you're old enough to have used a corded telephone, you will know that the cord is a helix or coil, and the more you twist the handset around, the more the supercoils form. More than that, if you have used corded phones for any length of time, you know that the more supercoils that form as you twist the cord around, the more the cord effectively becomes shorter and it fits into a smaller space. The supercoiling of DNA has been visualized by electron microscopy to show that circular DNA molecules really do take up less space when they are supercoiled. You can see here that the supercoiled DNA on the right is much more compact than the relaxed molecule on the left. So now let's think about the human genome. Each of your cells contains 6.4 billion base pairs. That is 1,380 times the size of the E. coli genome. And we know that the E. coli genome is 1.577 millimeters long, so that means the total length of DNA in each human cell is an incredible 2.176 meters, which is most likely taller than you. But humans, of course, are not single-cell organisms like bacteria. There are about 10 to the 14 cells in the human body, so that means that each of you contains over 217.6 billion kilometers of DNA. That's enough to go around the world 5 million times. Put another way, 217.6 billion kilometers is the distance that light would travel in 8.4 days through a vacuum. This is so much more of a problem than E. coli had to deal with, so we are past the point where simple supercoiling would do the trick. It turns out that eukaryotes like us have a much more complex solution to this packing problem. We start off, of course, with B-form DNA. Then rather than supercoiling, the first thing that happens is the DNA strand is wrapped around a special protein called histone to form a sort of beads on a string structure. A bead of several histone molecules with DNA wrapped around it is called a nucleosome. Then those beads are wound into a helical fiber about 30 nanometers in diameter. The fiber is then looped around in a sort of mini supercoil, and then 6 of these loops is arranged into a sort of rosette structure. Chains of rosettes are then themselves coiled, and here it is beginning to look a bit like the telephone cord I showed you earlier. And then just before the cell divides, when it wants to package the DNA in the most compact package it can, the coils are arranged into chromatids. Here we see 2 chromatids that are copies of each other just before they are pulled apart into the 2 daughter cells. This is 1 chromatid on the left and 1 chromatid on the right, and they are joined in the middle by what is called the centromere. We only call them chromatids when the cell is just about to divide, otherwise we call the individual DNA molecules that the cell holds chromosomes. And whereas E. coli has just 1 chromosome, humans have 2 copies of 22 chromosomes and 2 sex chromosomes, which can be 2 X chromosomes or 1 X and 1 Y. Individuals with XX usually present as female, and individuals with XY usually present as male. We call the human genome diploid because there are 2 copies of most of the chromosomes. Di is, of course, a prefix we often use to mean 2. Now we know that all these chromosomes represent a lot of information, but it turns out that there are specific sections of the DNA strand that encode for specific products in the cell. These are called genes, and I've highlighted a few examples here. A gene is a stretch of DNA that encodes the primary sequence of some functional product, which can either be RNA or protein, with a structural or catalytic function. Because there are 2 copies of most chromosomes, we also have 2 copies of most genes. but you will notice that XY individuals have only one copy of a lot of the genes on the X chromosome. In this particular bottom left example, the gene codes for a protein called glycogen phosphorylase that is expressed in the liver. So in the last few minutes of this video, we'll go back to the central dogma. As I mentioned in the last video, this is the idea proposed by Francis-Crick that there are certain pathways of information flow allowed in the cell. Copying of one DNA molecule to another is called replication. And although protein sequences are encoded in DNA, we can't go directly to protein, but must first copy information from DNA to RNA in a process called transcription. Finally, we can make a protein from the information on RNA in a process called translation. But I said before that dogma was not the right word for this concept because it had to be revised a few times based on new evidence. One of the pieces of evidence that really challenged the original central dogma concept is that some viruses have RNA genomes. You can see how proteins could be made from these genomes, but how does the viral genome get replicated? Well, this problem was solved by Howard Temin, a professor at UW-Madison, who discovered an enzyme that could replicate a viral RNA genome. He called it reverse transcriptase, for which he won a Nobel Prize in 1975. Retroviruses such as HIV have single-stranded RNA genomes, and during their life cycle they insert a copy of their genome into the infected cell's chromosome. But before that can happen, it has to be converted to a double-stranded DNA molecule. Surprisingly, all the steps necessary are carried out by one enzyme, reverse transcriptase. First, a complementary strand of DNA is synthesized using Watson-Crick base pairing. Then the RNA strand is broken down so that we are left with a single strand of DNA. And it turns out that reverse transcriptase can also make a second strand of DNA from the first strand, and this double-stranded molecule can then be inserted into the infected cell's chromosome. From there, the viral genes are transcribed and translated, and if we want more copies of the original RNA genome, that is just made through transcription of the whole inserted section. Now, one thing to note here is that human cells do not do any reverse transcription in their normal course of business, and so one strategy to treat HIV infection is to inhibit its reverse transcriptase, and one drug that does this is called zidovudine, or AZT. We'll go more into the mechanism of this drug during the nucleotide biosynthesis videos. So in light of the discovery of reverse transcriptase, we have to revise the central dogma to allow information flow from RNA to DNA, and we naturally call this reverse transcription. From subsequent discoveries, we also know that it is possible for some viruses to directly copy their RNA genome to make a new molecule of RNA. This is basically just like DNA replication, but with RNA, and it is carried out by an enzyme called RNA-dependent RNA polymerase, or alternatively RNA replicase. So the bottom line here is that I am showing the current iteration of the central dogma, and the pathways in blue are the ones that are exclusively used by RNA viruses. So at the end of the video, what have we learned? After going through this material, you should be able to understand how Watson-Crick base pairs determine the sequence of the second strand in a double-stranded DNA molecule. You should be able to outline why Watson-Crick's DNA structure explains Chargaff's rules. You should be able to explain the chemical basis for Watson-Crick base pairs, identify the handedness of helix structures, be able to understand the difference between major and minor grooves and how proteins interact with them, discuss the mechanism of anthracycline drugs. You should also be able to contrast the DNA packing strategies in bacteria and eukaryotes. You should be able to describe the chromosomes found in human cells and the significance of the sex chromosomes. And you should be able to identify the information pathways in the central dogma that had to be added to explain RNA viruses.